My name is Huseth. I'm an associate professor and extension entomologist in the Department of Entomology and Plant Pathology. I share responsibilities for extension and research with Dominic Reisig. Many of you will be familiar with Dominic from the grain circuit. I usually am doing quite a bit of extension work in sweet potatoes as well as cotton. So you might have seen me speak at some of those events, but I also do a lot of work in grains. And so today, the charge was to think more broadly and deeply about some of the insect-related issues that are problematic for North Carolina soybeans. And I was asked to talk a little bit about stink bug management. I added another word in there, 2 words, ecology and management. I think that this is a really important concept to consider when we're trying to make good management decisions about stink bugs. You know, what are they doing in the landscape throughout time, throughout the production season, and even into overwintering, can give us a good idea of where we expect risk for infestations to occur in soybeans, corn, cotton, etc. So the goal of this talk is to think a little bit more like a stink bug. You know, where we go in the landscape to find these insects, how we scout them. A lot of that has to do with that E word. And so we're going to talk a little bit about ecology, do a little bit of a deep dive, and think about stink bugs in North Carolina. I'd like to pause for a second and really thank my graduate student, Sujin Panta. He's a PhD student in the lab. He's done the bulk of this work. So he's been out throughout North Carolina sampling stink bugs in soybeans, in wheat, trying to bring more understanding to the places that we tend to find these insects across the eastern coastal plain through to the mountains. So the outline today is really to think about that ecological principle first. That informs how we think about management later. So just what biology of true bugs is, how they reproduce, what we can expect for their life cycle. You know, what stink bugs are economically important in the state of North Carolina. We've seen a shift in the species composition over the past several decades. So I'd like to bring that in, you know, what to expect in the future. A little bit about where we find stink bugs in the central coastal plain. So we've done a lot of work in counties surrounding Wayne County, but, you know, bringing to bear like what to expect. And then some really practical information about what we're seeing as far as resistance to bifenthrin in particular in brown stink bug populations. This is a major concern because we feel like we're using bifenthrin, particularly in this central coastal plain area in corn. And so what's the expectation that resistance will evolve, knowing that bifenthrin and other pyrethroids are really critical to managing both corn, stink bugs in corn, cotton, and soybeans. So we really wanted to keep track of that. So just getting right into it, true bugs. So I'm an insect, sort of an insect nerd. It's important to remember, you know, that these insects are part of a very broad group of arthropods called true bugs. They are diverse, and you'll see a lot of pest species in here that are important both for human health as well as for crop health. So if you've ever had these guys, you probably remember it quite distinctly. Bed bugs are a major problem. They're all in the same group. Here's burrower bug. This is an emerging pest of peanut. We're trying to keep track of that issue, but you'll see there are several other species in this group. We'll focus on stink bugs today, but they share a very general life cycle in which we can expect predictable patterns of development and ecology. In addition to the pests, we also need to think about the beneficials. And so if you're a cotton grower, oftentimes we're thinking really intensively and deliberately about when we're positioning particularly broad spectrum insecticides with respect to bloom periods, knowing that several of these insects, these true bugs, particularly minute pirate bug and big-eyed bugs, are major predators of earworm eggs in fields. And so we know if we spray something like acephate pre-bloom in cotton, we can create a huge bollworm issue just as a result of eliminating natural enemies. The same goes for for soybeans, where we know that, you know, using some of these heavier broad spectrum insecticides can cause more problems for things like earworms later in the season, particularly at the reproductive stage. So it's important to consider that not all true bugs are bad bugs, but we also can get a lot of benefits from these natural enemies. Much of this is difficult to quantify. But certainly choosing selective insecticides can be really important for alleviating any undue pressure on these populations. So a little entomology 101. What is a true bug? They have a very characteristic body layout, and this includes stink bugs and also tarnished plant bugs, so a very common pest in cotton. They have this sort of membranous hind wing and a forewing we call a hemielytra. Basically, they have this very characteristic layout, but this sort of area is very characteristic of many of these true bug species. So whether you're looking at a ligus in a field or a tarnished plant bug in a field or a stink bug, you're going to see that sort of layout. Oftentimes our stink bugs have this very characteristic sort of pentagon sort of shape with a broader area in the front and then trailing into this membranous area. They all have tubular mouth parts in which they're feeding on plants. And so that's why it's really important to think about they're difficult to assign as far as feeding injury in a lot of cases because they're not like a caterpillar that's actually consuming a leaf or a boll. They're piercing into the plant. They ingest saliva into the plant part. Oftentimes that creates a plant defense cascade, and that's where we see injury and yield losses. And so thinking about these stink bugs, particularly in soybeans, feeding through that pod wall and causing those reductions in seed quality is really important. So there's been a lot of work thinking about how purple seed stain and other factors are related to stink bug feeding. That's one part, because it's not that the seed isn't there, it's that they've fed through the seed wall or the pod wall into the seed and cause yield reductions and also quality reductions. So the general life cycle is as follows. Certainly you might see some of these. These are the immatures. So these true bugs look essentially like the adults. The one distinguishing difference is that these guys are not reproductively capable and they don't have wings. So the immature life cycles are similar to the adults except for the wing patterns, and they go through these different instars, and then they become a reproductively active adult. The nymphs, particularly in stink bugs, can be at various colors that are different from the adults. So some of the southern greens are very, very showy. They have different coloration than the adults. But the important distinction, both nymphs and adults can feed on plants. And so when these insects immigrate into the field, they lay a bunch of eggs. These eggs eclose. As soon as those eggs are hatched and they're feeding on plants, they become a concern for farmers. So it's important to understand that this is where we get into that transition from biology to ecology, thinking about where they are in the landscape. Stink bugs overwinter as an adult in general, mostly in duff layers, in forests and places like that. So very protected areas in which they can withstand winter lethal temperatures by having some moderation of and insulation from what's happening in the ambient air temperatures around them. Stink bugs, are triggered into what we call diapause, which is just their overwintering stage, by a combination of photoperiod, so changing sunlight conditions as well as temperature. And with an increase in temperature, these insects emerge. And so as we move into February, March, and April, there are a lot of weedy hosts that these insects will emerge, lay eggs on, and start their life cycle. One major host that we know of is certainly wheat early season, and a lot of growers have picked up on that, is that where you have wheat adjacent to corn is oftentimes places in which we get stink bug infestations along that wheat-corn interface, and I'll talk about that in a little bit. So they go through a cycle on wild hosts. They lay all their eggs in the crop. Certainly we see these very characteristic aggregations of stink bugs maybe at the top of the plants. And then it takes them quite a bit of time to develop. This is not as fast as, let's say, an aphid or a thrips. It takes egg to adult about 36 to 45 days. So they're in the crop for quite a long period of time, which is important. Important to consider is primarily because we know that there are very discrete periods at which the crop's economically susceptible. Reproductive soybeans is one period that we're really concerned about stink bugs. but these insects can be present in the crop at vegetative stages or even after R6 when not causing a lot of economic injury. So it's the alignment between the susceptibility of the crop and the pest that's the important thing to consider. What we do know about stink bugs is that local movement can provide, some important insights into where we expect infestations to happen. And so this is kind of getting to that part about thinking like a stink bug. I'm going to walk through the cycle of stink bug populations generally in this hypothetical landscape. And so here's a aerial imagery of a farm or a set of fields adjacent to a wooded boundary. We know that brown stink bugs and green stink bugs tend to overwinter in some of these protected areas like forests. So right now they're hanging out in this forested area. They're down in that leafy litter, in pine needles, etc. Then they subsequently move into their next host. So those stink bug populations will move into a very suitable early season host. One that's really important is early season wheat. They'll conduct a full generation in that wheat field, meaning that a female can lay several eggs in the 10s to 50 eggs, and then they close. That immature population completes development. Maybe you go in and pick that wheat field, anticipating planting it back to soybeans and double crop, but they choose to move to an adjacent full season soybean field. And so this is where we get big pulses of these insects into an adjacent crop. One great example is corn, corn or soybeans. Along these edges where they've moved from the wheat field into the next suitable host is where we expect to see our highest presence of stink bug populations before they dilute out into the rest of the field. And so these are places in which you can target scouting to try to understand and use that landscape to your advantage, knowing that there's a lot of stink bugs that are likely coming out of that wheat field and moving into the next suitable host. So thinking more like a stink bug is important here, and thinking about places in which you might need to scout more diligently, particularly in something like full season soybean or corn. Later in the season, after that crop senesces, these insects move on to weeds. They'll feed on that weedy host, and then eventually they immigrate back into that forested habitat to overwinter again in the season. So this sort of local movement pattern is really important to consider as we try to target stink bug populations. Knowing the places where they move through the habitat or through the landscape is critical for being able to accurately scout. So now I want to transition. This is much of Sujin's work, just thinking about places that we expect stink bugs to be in North Carolina. And this is essentially what we did in a nutshell. We don't need a lot of bar graphs or anything like that to communicate how we did this study. We basically have an army of technicians and undergrads that go out just like an independent crop consultant would and scout soybeans across the state. And why is that important? Unlike a lot of very focused, replicated studies, my program does a lot of these sort of on-farm projects where we're trying to collect as many fields as possible and stink bug data from all these different fields and put it all together to tell a big story about what is the abundance of stink bugs, what species are common, with the goal of being able to assign risk estimates for different counties, what stink bug species are present. And you might ask, why is that even really important if what we think about is a field-by-field basis? Why it's important is we do know that several of these stink bug species respond differently to different insecticides. So we know that brown stink bug is less susceptible to some pyrethroids than others. So understanding what the likely species composition is going into a season can help inform the type of products you might want to have on hand, say if you're in a heavy brown stink bug environment. And so together with on-farm research that's very focused on efficacy, yield, and everything like that, this is a complementary piece of data that can help inform larger scale decisions. So it's important to consider why stink bugs have become such a problem as well. So here's basically in a nutshell the reasons that we think stink bugs and tarnished plant bugs have become such a problem. Since the Green Revolution, we've really had an increase in broad spectrum insecticides and now a decrease. We have more specific insecticides in the mix targeting our key pests. We have eradicated boll weevil, one insect that was, sprayed widely in cotton. A lot of the sprays that targeted boll weevil also likely suppressed some of these true bug populations. And at the same time, since the mid-90s, we've seen an adoption of transgenic crops. What the result is there's essentially less insecticide in the system and more favorable conditions for secondary pests. So we think that this is one of the major motivations for why we've seen this rapid increase in these true bug species across the southern US is a combination of several of these factors and probably others that contribute to an emergence of some very difficult pests to control. In comparison to something like boll weevil or budworm, bollworm, we're probably in a lot better place controlling stink bugs, but we do know that sprays continue to increase. These insects continue to become a challenge in our production system. And so we think about basically two of those secondary pest groups, the tarnished plant bug, this is mostly a cotton pest, but then in soybeans, this whole complex of stink bug species. So brown, green stink bugs, southern green stink bugs, brown marmorated stink bugs, and a host of other ones we'll find commonly in soybeans. So we think about, what's the economic impact of these? This is where the rubber meets the road from a regional perspective. Stink bugs are an important pest of soybeans, particularly at the reproductive stage. Here's some immature green stink bugs. As you can see, their coloration is very different, as I was pointing out before. Brown stink bugs, brown marmorated stink bugs is a pest in the mountains mostly, and southern green, or green stink bugs, excuse me. We've found that in soybeans across the US, 28% of the combined insect-related losses are attributable to stink bugs, with a major emphasis on brown stink bug, green stink bug, red-banded further south. But what we've found here in North Carolina is southern green stink bugs, because of a variety of unexplained environmental conditions, likely, we are seeing an increase in southern green stink bugs. They're both earlier and more abundant in our system. So there's something going on that's really favoring southern green stink bug infestation. So it's something we're keeping track of, part of the motivation for this work. So what I always think about is, where have we been and where are we going? And again, most of this motivation for this work was driven by this, which was an arthropod survey in soybeans, so many, many insects, conducted in 1973. So researchers at NC State, many of you will recognize names like John Van Dyne, J.R. Bradley, maybe Bob Rabb. Folks that were in the Department of Entomology got funding to look at what insects are commonly found in soybeans in the 70s. Fast forward to 2023, the question became, well, if they found that these insects were common in different places in the 70s, are these same species as abundant in North Carolina soybeans right now? So it's a good baseline assessment of where we might be and where we might be going fast forward several decades. So here's data from that. Here's a line chart. Here's Edgecombe County. Here's Washington County. Here's Johnston County. They found that, stink bugs on average were fairly low. There were some cases, particularly in Washington County, in which they found high abundance of stink bugs. But I really wanted to know, given this information from the 70s, how has it changed over time? So this is what we did. This is work funded by the Soybean Producers Association in particular. And our goal was to revisit that area-wide survey, expand it to the mountains as well as the Piedmont, and ask the question of, in reproductive stage soybeans, what is the abundance of species? And so what we did was we sampled about 30 fields per year from 2022 to 2024. So we're going to have about 90 fields across the state, and we added another 30 fields from Virginia. We swept R3 to R6 beans. we looked at each location. So we sampled the perimeter of fields. Again, this is where we expect the highest densities of these insects to occur. And we asked the question of how abundant are they at the field level, total, average, or at any individual spot. So this is like a twofold kind of study asking questions of variation inside the field that gets at scouting questions, but then also these sort of broader geographic questions about what insects, what stink bug species and natural enemies occur in soybeans right now. So this is our progress. Our stars are what we're doing in 24. We've distributed our sampling across ecoregions. So these are just very understandable areas, the mountains, the Piedmont, and the eastern coastal plain. We sampled all of these fields in 2022 and ID'd all the insects that came out. We're going to talk a lot about this central coastal plain data today, but bear in mind that we're making comparisons from here to here and trying to understand how variable these populations are. What I think the benefit of this is once we kind of complete this central coastal plain map for growers that are in the room, we're going to be able to deliver a fairly comprehensive set of species IDs and things to expect, as well as some refined recommendations for distributing scouting as a result of what we've learned on a field-by-field basis. So right here, here we are in Goldsboro. As far as 2022 and 2023, this is our central coastal plain sampling. I'm going to present that data. This is what we think of as stink bug central. So we reasonably thought that we get a lot more pressure out in the Washington Blacklands area. It's concerns by corn growers in particular have really defined that this area, the central coastal plain, due to these fragmented small fields likely, are places where we find a lot of stink bug pressure. So this is a great topic for this audience. And we've been to all of these dots and really thinking about these 14 fields in Sampson and 9 and Johnston, trying to find out who's most prevalent. So across that Sampson County data set, this is relative abundance. So if we added up all the stink bugs we collected and figured out what proportion each species occupied, we found by far and away green stink bugs were the most prevalent species, followed by brown stink bug and then southern greens. We have many minor species and no brown marmorated stink bugs. So this is a little bit of a surprise. We expected to find in Sampson County more brown stink bugs in soybeans, but it gives us a little snapshot of what species to expect. Why this really matters is, these insects are recommended to be, we recommend bifenthrin as well as to a lesser extent acephate for control. These are much more susceptible to other pyrethroids. And so knowing who you have in your fields is really important for making good insecticide decisions because all these products are not created equal. In Johnston County, we had 2 years of data. Again, it's the same layout. We saw more brown stink bugs in 2023 than green stink bugs. We saw the reverse in 2022. So there's a lot of local variation happening in this system. Field-to-field variation is quite clearly really important. And the composition of stink bug species seems to vary considerably among fields. And so that's really important to think about as we're making scouting recommendations, as well as the kind of crop reports you might get from your independent crop consultant, or if you're scouting the crop yourself. It's really important to be able to distinguish these different species. So when we break it down, basically our top winners for Sampson County were green stink bugs, southern green stink bugs in Johnston County as far as the percent abundance across these different sites. And what I'll note that percent abundance is rather deceiving because these numbers are quite low. So we sampled about 250 sweeps per field. So the economic threshold, which I'll get to in a second, would suggest that we're far below economic threshold in many of these instances, but it does give us an idea of the relative abundance of species across these different locations. So economics. this is one piece of the puzzle. This is the prevalence of the insects, or at least the abundance of the insects from our sweep samples. So our sweep economic threshold in North Carolina is 5 stink bugs per 15 sweeps. These are all the fields that I presented in the prior slides. So we had a map of these fields. Here they are just broken out categorically. So each independent field just got a number, and then we ordered these values based on their average. And so the dot is the average per 250 sweeps. So basically our stink bug threshold, so it's about a little bit north of 2.5 insects per 15 sweeps. So that's below our economic threshold. But what's important is the variation. So as Rod pointed out, variation is part of the puzzle, not just our average. So while our average is below the economic threshold, if you happen to jump out of the truck and sample one corner of the field and you found, whoa, I'm way above threshold, it's not the whole story, right? And so making good decisions about what the average is important. So this is the average among our 10 sampling spots, et cetera, boiled down into our economic threshold. But it's important to note that you could either make a bad decision and miss a bunch of stink bugs, or a good decision and detect stink bugs, but really accurately sampling across the field is going to be important, which is something that as Rod pointed out, insects, nematodes, diseases are notoriously spatially aggregated. And so it's hard to sample one spot and assume that it is representative of the whole field. Really, we need to get a good representation across space. We saw the same thing in Sampson County, where our highest max value per 15 sweeps was also the field with the largest variation. So these things are challenging to scout. Unlike pests like earworm that are sort of uniformly distributed in the field, stink bugs, plant bugs, et cetera, are a major, major challenge. For this reason, is that small sample sizes can really complicate our interpretation of whether or not we're going to make a spray decision. So if you are scouting, this is our recommended thresholds. You can neglect this seed and edible side, but certainly for grain, depending on the row spacing, basically we're thinking 5 per 15. If you happen to use a beet cloth, the numbers are quite a bit smaller. And so this is where we make the economic decision about, or the insect side of the economic decision. Dr. Pickett's going to talk about the economics of soybeans later on. That's the other piece of the puzzle about making a decision. What's your pesticide cost and what's the value of beans? So remember that. This is a good guideline for making an economic decision based on the insects. And just in conclusion, I wanted to touch on pyrethroid resistance. So this is a huge concern that we had. We didn't have any baseline data. Researchers in the Mid-South have reported pyrethroid resistance in several different species going back into the 80s where they're intensively sprayed. We knew that there was a lot more pressure being put on brown stink bug populations as a result of a lot of reports of poor control in corn, sort of that banana ear, and that motivated a whole bunch of work on the corn side. But we did know that we're spraying a lot of brown stink bug populations in corn, probably in cotton, and also in soybeans. And so we wanted to get a beat on whether or not there was any suggestion that there was pyrethroid resistance, particularly by bifenthrin. So resistance, what is it? Here's a graphic I made. I think it communicates what we're doing here. Remember, we have just this huge population of bugs out there. In any given population of insects, there are going to be resistant individuals. It's a small fraction of the population, but they're out there. So you bring in a new insecticide, let's say when we released Brigade or bifenthrin decades ago. Really what it does is it knocks out all the susceptible individuals. If you repeat and use that same insecticide time and time again, what the end result is you get a big population of these red guys, which are our resistant bugs. So what we want to do is break this cycle up by rotating insecticide modes of action. Much like the plant pathologists will talk about rotating modes of action for diseases, we encounter the same challenges for insecticides. This repeated selection is what drives the favorability for resistant individuals to emerge, reproduce, etc. Screening, which is what I'll present next, is our useful estimator for where we expect a population to be. So we take samples of populations, we bring them into the lab, we expose them to the insecticide, and we ask the question of how many of the population sample dies. So it's pretty easy, but really what we're thinking about is, can we measure a population and does it look like this, or does that population look like that? And we collect insects from across the state. We ask the question of, does Sampson County look like this? Does Wayne County look like that? That helps us inform you about products to choose so that you don't have crop failure. So basically what we did is we have a glass vial, we make up an insecticide solution, we put it on our favorite tool in the lab, which is a hot dog roller. Same thing you would find in a gas station in eastern North Carolina. We let the acetone basically volatilize off, and what's left is a residue on the inside of the vial. We collect stink bugs, we put them in the vial, and we leave them for a period of time, and we ask the question, did they die or did they live? So if you do that on enough bugs from enough populations, you start to get a picture of what the range and susceptibility is. And if we happen to find a hotspot, say Clinton in Sampson County, we can inform growers in that area that they might have a problem. They might choose a different insecticide than bifenthrin. So to date, we've sampled and characterized resistance in several populations of green stink bugs, brown stink bugs, and southern green stink bugs. These insect collections are generally done when we get a report from a county agent that says we've got a problem. We're either collecting them from wheat or soybeans, but I think that this is the most important thing. So this is our estimator of resistance, and what we see is that the lethal dose is far below what we would expect based on prior published literature from the Mid-South. So this slide, even though it's a lot of numbers, my interpretation is we seem to be in a reasonably good place for bifenthrin resistance in stink bug populations we find in North Carolina. So that's important. It doesn't mean that we've sampled every population, surely, and resistant insects definitely exist out in the landscape, but we have not found them yet. So if you happen to have a crop failure or a spray failure in either corn or soybeans, reach out to your county agent, reach out to me directly. We would love to add to this slide, get more relevant information for producers, particularly in this county area where we know we're using a lot of these products. So if you have a stink bug problem, let us know. We'll come out and make a collection and generate on-farm data for you. So just in conclusion, part of the puzzle of this is choosing what to do and when. Our insecticide recommendations are published annually in the Ag Chem Manual. It's a great resource for disease management, nematode management, insect management, fertility decisions, etc. What we do know is that reading that product portfolio can give you an idea of what stink bug species you have and what we would recommend. Certainly we know that green and southern greens are easier to kill with some pyrethroids. Brown stink bugs require higher rates, something on the order of 6.4 fluid ounces of bifenthrin or acephate north of a ½ pound. Are all good decisions for brown stink bugs. So knowing which species you have is critical. Product recommendations, certainly can be found here. It's available online. We're working on an app that's going to be updated as far as crop protection tools that you can access more easily than the PDF online. So stay tuned for that. With that, thank you for your attention. There's my contact information on your left, Dominic's contact information on the right. I'd really like to thank the Soybean Producers Association for funding my program to do a lot of this work. Certainly the NCE Plant Sciences Initiative for supporting my program. That's where we're housed, and ongoing work there. And with that, I'd be pleased to take any questions you might have.
Yeah, would you mind making a comment on the smart traps?
Oh, okay. So Jeff Chandler asked about our smart traps for corn earworm. So There's a little bit of backstory. If you've seen the cotton production circuit in years prior, we've developed in collaboration with engineering, electrical engineering at NC State, a way to retrofit a standard corn earworm trap, which are very common. We probably have 50 of these traps in our shed. We basically 3D printed an IR sensor on the top. The simple way to think about that is it's basically a burglar alarm for an earworm. So as soon as that earworm moves through the trap, it makes a count. And that trap sits out in the landscape, and basically what it is it's making that count. It's got a weather station on it, and then in this year it's got an LTE chip, just like in your cell phone, that will uplink the data to the cloud. And basically what we're moving at this is we wanted to produce an inexpensive trap through the public sector that we could produce and put in the hands of growers for less than $500. We're thinking it's about $150 right now. We're in the process of thinking about how we're going to commercialize. But it's a really useful piece of information that provides you day-to-day updates on pest capture. We're going through the refining process of it this season. We put out 14 traps on a lot of different acreages in a 5-county area north of Wilson County. And so I think in 2025, we're gonna have a product that we're gonna be able to deploy more widely. So where we're thinking about putting this is through the portal. So for producers that have access to the cotton portal or the trap portal database, you're gonna get more updated data and then pair it with some data analytics to do some more risk forecasting for earworms. So it's an ongoing project. Thank you for the question. I think we're in a good position to get that done. Sir.
Have you seen any issues with spraying pyrethroid or any other insecticide in combination with fungicide and or sequential herbicide, prefix or Roundup?
Okay, so the question was, is there any antagonism between common insecticides, namely pyrethroids, and a herbicide like Roundup or a fungicide? So admittedly, entomologists, plant pathologists, and weed scientists, we don't get together very often and do that study, but it's needed. Just like we get a lot of questions about adjuvants, that compatibility piece is really important. We don't generate that data, but the best place to go is on the label. So any insecticide that has any suspicion of having antagonism issues, be it the formulation, it's not compatible, maybe it's tank pH as a result of whatever product you're mixing it with, should have a warning on it. But unfortunately, we don't generate that information as frequently as we should. But good question. All right, thanks.